I'd like to introduce Anna Gruvstad Melén, who graduated from CNELM with the MSc. Anna is the co-founder of a functional medicine clinic in Sweden, specializing in ME, CFS, and mental health conditions. Alongside clinical practice, Anna is a research supervisor and tutor for evidence-based personalised healthcare at CNELM and a board member of MTech. Anna's presentation today is on her dissertation, the impact of personalised nutrition on mitochondrial function in ME, CFS, a retrospective quantitative study.
Thank you.
Welcome, Anna. Hi.
Thank you. Okay, so there are different research areas at CNELM that we want to focus on, and my research falls under number 4, which is statistical analysis of laboratory tests. My research was inspired by the methodology of Myhill, Booth, and McLaren-Howard's study from 2013 of the effect of a personalized nutrition intervention on the mitochondrial function of ME/CFS patients. And the aim was to develop statistically sound mitochondrial function measures, so-called components, based on their panel of mitochondrial function markers. We wanted to determine whether these measures could be used to predict pre- or post-intervention status, and to evaluate whether these measures could have a potential to be used as outcome markers in ME/CFS. And the steps to do this, to statistically analyze retrospective pre- and post-intervention data, was by using principal component analysis to identify multiple measures of mitochondrial function based on the markers identified by Myhill, Booth, and McLaren-Howard in 2009. I will show you those markers later. And then the second step was to use logistic regression to assess the predictive capacity of these measures to determine whether a given data point was a pre- or a post-intervention. I also did a literature review before the quantitative study to critically investigate the current evidence of mitochondrial imbalance in ME/CFS. And the main conclusion from the 45 papers, of which the majority were case control studies, were that mitochondria play a significant role in the pathophysiology of ME/CFS, and that they're less adaptive to environmental stressors. One of the main studies were Navio et al. From 2016, who did a case control study. I would say this is a groundbreaking study. They proposed that the illness is an evolutionary defense mechanism against endogenous and exogenous stress triggers, trading function for survival. They also demonstrated that when mitochondria are under stress, they can leak ATP outside the cell, where they function as danger signaling molecules to alert the immune system. I also found evidence that measuring ATP and OXPHOS is not enough to detect mitochondrial dysfunction in ME/CFS. And this is really important because many authors in their studies, they just measure total ATP. So what is important is that the efficiency of the translocator proteins must also be taken into consideration. The transporter proteins, they import ADP from the cytosol into the mitochondria, and they export ATP from the mitochondria to be used for energy. I also found an evidence of a correlation between low levels of CoQ10 and NAD and ME/CFS. This was found in several cross-sectional studies and in one systematic review. I found that oxidative stress was the main mechanism involved in mitochondrial dysfunction within the review. And some of the reasons for that could be due to oxidative stress from virus, bacteria, or other potential triggers such as environmental toxins that inhibit processes in the electron transport chain and then inactivate mitochondrial enzymes depleting ATP, for example. I also looked at 12 human intervention studies, and found that CoQ10 and NAD together were the most effective intervention reducing oxidative stress in ME/CFS. Okay, so the motivation for this study from the beginning was that I found that I thought that the ATP profile test was unrecognized and underappreciated as a diagnostic tool for measuring mitochondrial improvement in ME/CFS. The test was developed by John McLaren Howard during his career as a laboratory director at Biolab, and later it was carried out by himself in his own lab in Acumen Labs in Devon. So the background literature were 3 studies from Myhill and colleagues. The first one, they demonstrated a correlation between the clinical disability of 71 ME/CFS patients and their mitochondrial energy score compared to 53 controls. Their energy score, you can see here, it's 5 different factors that they multiplied together to get this energy score. And this score is the one that we wanted to improve with our study. The study in 2012 explored some of the mechanisms in ME/CFS and concluded that the reasons for mitochondrial dysfunction were either due to substrate or cofactor deficiency or due to inhibition by a toxin. In the latest study, and the study that we wanted to reproduce was an audit of 34 ME/CFS patients who also did a follow-up. It was 30 patients who followed the regime, and they all improved their mitochondrial function. And the author said that improved function tests are usually paralleled by clinical improvement as well. So looking at the PMO framework, the population, we looked at 71 patients with ME/CFS from the Optimum Health Clinic, which is a private clinic. They were between 15 and 70 years old. The intervention was personalized nutrition interventions recommended by nutritional therapists. The mechanisms were mainly mitochondrial dysfunction, but also oxidative stress. And the outcome measures were components obtained from the ATP profile test. So looking at the ATP profile test markers, they're categorized into different groups. First, you have the ATP studies done in mixed leukocytes. So you have ATP in whole cells, ADP to ATP conversion efficiency in whole cells, translocator studies in mitochondria, and there are other assays as well that we looked at. So you have cell-free DNA, and this can be released due to apoptosis, necrosis, or netosis. Cell-free DNA seems to increase reactive oxygen species, so this is also an indirect measure of oxidative stress. And in vitro studies have shown that there's evidence that cell-free DNA is a stress signal released in response to oxidative stress. And then we have the SOD studies, which are like the oxidative stress markers. This is just for your reference. You can have it the slides later. I'm not going to go into every marker, but there are 18. So we did a principal component analysis, and this analysis yielded 4 components with an eigenvalue above 1. And as you see, these components explain 81.8% of the variance. And what this kind of analysis does is finding markers that go together. So the so-called loadings are also the weightings that would be used to get a score for each patient. The highest loadings are the most relevant markers, and you can see that they're listed in order of significance. And correlations lower than 0.4% have been blanked out because they're probably not significant. We also did a logistic regression model, and you can see that 89% of the time the model is correctly predicting whether a data point is pre or a post point, which is quite good. If we look at the statistical significance of the components, factor 1 and factor 3 are statistically significant, while factor 2 and 4 are quite borderline, but we don't want to exclude them anyway. It could be a sample problem, so it would be interesting to do with a larger sample. Just for you to see visually how the data points are moving from pre to post. You can see in the first scatter plot that they're kind of two distinct clusters, and then they move to a more homogeneous group in the post group. We don't really know exactly what this means, but we see that there is a big difference. So, conclusion. Can we use the ATP profile test as outcome measures? We would say yes. Our model was highly predictive. We also have some supporting literature from Arrow and Howard from 2012. They did a preliminary prospective study with 3 groups, psychology, nutrition, and combined, and they all made significant improvements clinically. And considering that the recovery rate of ME/CFS is estimated to 5%, any kind of improvement is really good. We have the MyHill study from 2013. Then we have some contradictory literature as well, because just as I had handed in my dissertation, Thomas et al. Published a paper where they demonstrated that there was no difference in ATP profile tests between ME/CFS and controls. But looking at their paper, they had a very small sample size, and they didn't look at the translocator studies, which we found really important, and so did MyHill. They also didn't replicate the MyHill score, which was the aim of our study as well. Sorry. Yeah, okay, so the limitations. We only did a pre-post observational study. We didn't have a control group, no inclusion-exclusion criteria, no information of how many patients were enrolled in psychology treatment, and no information on clinical improvement. The strengths were the sample size, 71 patients, which is quite big for a ME study. The length was 3 months to 5 years, with several follow-ups. We had rigorous statistical method and a new score with weighted sums, which simplifies interpretation of tests for NTs, and a much more robust way of understanding mitochondrial dysfunction. So this is the last slide with future research. There are many gaps in the ME/CFS research out there. They use different study designs, diagnostic criteria, measures of fatigue, symptom scales, etc. It would be interesting to do a muscle biopsy and compare with the ATP profile test, do maybe case control RCTs, measuring baseline cofactor levels, oxidative stress markers, taking an ATP profile test, then intervention with cofactors and retake, for example. I don't have to go through all of these, but also it would be interesting to compare with a group of depression and anxiety, because there are also studies showing that they have mitochondrial dysfunction. What we want to do at CNELM now is to publish this study. We want to also publish a review study on oxidative stress in mitochondrial dysfunction and the nutritional interventions that we looked at. We would like to do a case control study, including clinical improvement. And perhaps if we don't get like a perfect separation with the data, we could use more sophisticated machine learning approaches, such as kernel machines. Thank you.
Thank you.